So why is sample quality control so important? So first of all, it's a validation that your sample preparation worked as planned, and so that you're sure that the samples you provide are as good as you can make them. And this is, of course, necessary to get good and reliable data for your experiment. And it's also necessary to measure the samples to be able to adjust the amount and concentration to fit the requirements of the selected library preparation method. And some samples are, of course, more challenging than others. But by knowing this already from the start, that makes sure that we can maybe be prepared for this and possibly enable us to adjust preparation parameters or maybe recommend another preparation method that's better suitable for your samples. So we offer quite a lot of different library preparations, and most of them are validated and used routinely in our production environment. And we also offer some other more experimental protocols for selected projects. So here is a visualization of what we have. So for the standard library preparation, we use the TrueSeq PCR-free kit that Pavel described. And we also have a non-Illumina kit for more challenging samples, where we can start with sub-nanogram amounts of DNA for various applications. We also do targeted enrichment using the Agilent SureSelect kits, and finally also mate pair libraries using the Illumina Nextera mate pair kit. And for RNA, we have basically 3 different tracks. We use the standard poly-A selection, also ribosomal depletion using the Ribosero kits from Illumina, and we can also do the small RNA preparations. And all of these different preparation methods have different requirements, so it's important to make sure that your samples are suitable for the selected method that you want to use. Quality control is difficult. And we handle thousands of samples every year, and making quality controls also takes a substantial amount of time. It's both lab work, administration work, and also a lot of communication back and forth between you and us if there are problems with the samples. And we also want to handle the original sample plates as little as possible to avoid risks of cross-contamination and so on. So this is the reason why we want to avoid diluting, concentrating, or pooling samples. So we just want to focus on the things we're good at, library preparation and sequencing, and so we can deliver the data as quickly as possible. So this means that we want you to make sure that the samples are as good as you can make them and fulfill their requirements when we get them. So this is a brief overview of kind of a project startup workflow. So if possible, it's always good if you can discuss the project with us before you start the experiments, and also discuss it with the bioinformaticians involved, because we might suggest alternative preparation methods and so on that might be better suitable for your experiment. You should, of course, measure and evaluate your samples and possibly adjust them to the requirements of the selected method. And when this is done, you fill in all the sample information in an Excel sheet that you get from us. You send this together with maybe additional information such as gel pictures and so on. And we review this to make sure that everything looks good, so you can possibly make adjustments to this before the samples are submitted. We also do quality control of the samples when they get here, but that's mostly to validate your measurements and make sure that all the samples that we take into our system are measured using the same standardized methods. And if there are any deviations from the requirements, we will contact you. And this process can take quite a long time if there are problems. First, we have to do the measurements, and then we must contact you, and maybe you need to redo more preparations and so on. So it can be quite time-consuming, and the project doesn't get queued until everything has been settled, that either you have produced new samples or you want to continue with the samples as they are. And these sequencing projects can be quite expensive, so it's so the quality control is also a way of establishing a mutual agreement between us and the user to establish who should take the economical responsibility for the preparation of the samples. We have a rerun policy that is based on the QC status of the samples. And we will do up to 2 repreps and up to 2 sequencing starts for all the samples that pass the QC criteria. And here I'll try to illustrate this. It might look a little bit complicated. So in the vertical direction, it's the different QC steps involved in a project. So at the top, we have the sample QC, where we check the amount and quality of the samples. Then we also do a library QC to make sure that the libraries are good for sequencing. There is also sequencing QC, where we look at the amount of reads produced for each sample, as well as run parameters such as error rate and total yield and so on. And lastly, there's also bioinformatic QC, where we analyze the data to check for duplicate sequences and so on. So if the samples pass the sample QC, it's all okay. We will do a reprep of the samples if they should fail for some reason in the library prep. And if the second reprep doesn't work for some reason either, you do not have to pay for the library preparation of that sample. However, if the sample should fail in the reception control, we will contact you and ask if you want to replace it, or if you want to take a chance and go on with the sample. Anyway, it might be a rare sample and you're not able to make any new extractions. In this case, for failed samples, we will do no additional attempts if the first library preparation should fail. And you always have to pay for that library preparation, even if it doesn't produce any usable library. But since I mentioned before, quality control can be quite difficult. So it happens that failed samples produce seemingly good libraries that pass the library QC, and also the other way around, that samples that pass the quality control might fail both attempts at producing a library. So if the library preparation fails, if you want, we can try anyway. We can, of course, make a prediction of whether it's likely to work or not. But we will do no resequencing attempts if it should produce bad data or too little data and so on. And also a little exception here is that if a failed sample should produce a, sorry, yeah.
Quick question. What is the failed library? What would be the lowest concentration that still can be sequenced?
Yeah, so we have some internal criteria here where we look at both the concentration of the sample, but also the size distribution, if it's too big or if there are primer dimers and so on. And it's a little bit different criteria for high-seq or MiSeq. For high-seq, we want a final concentration for clustering with about 2 nanomolar, I think, and I think it's a little bit more for the MiSeq. Right.
Would you still sequence if there was a bit lower concentration?
We could try. I mean, it's of course not a clear cutoff there, so even lower concentrations can of course produce data. So it's kind of a we have to discuss this basically individually for each case. Right. Yeah. So if a failed sample should produce an OK library, if it should fail the bioinformatics QC, where we check for duplicates and so on, that will not make that we do a rerun, because this might be sample related. It could be too little sample, for example, or bad quality that produces a lot of duplicate sequences and so on. So we quite often get this question: why do we need to measure our samples when you do it anyway? So, and this is something I've tried to summarize now over the previous few slides, and it's essentially a combination of efficiency and quality. So it saves time for everyone if the primary quality check is done by you, and our additional check is more to verify that the values are correct using a standardized method used for all of the samples that we run. And how do we define these requirements then? So all new methods are validated to work with the instrumentation available here in our lab. And we also want them to produce reliable results for a wide variety of samples. So this could be the explanation why our requirements are a bit tighter than the requirements specified for the kits that we use. So the volume and concentration of the submitted samples must be compatible with our liquid handling system, and also the QC measurement methods. And the amount must also be enough to reliably produce enough good quality library for many different kinds of samples. And requirements are also changing, especially when we're breaking in new methods. So it's important that you read the sample information that we send out to you when you start a new project. And if you have questions, you should always ask us, and make sure that the information you have is up to date. So during the sample quality control, there are these 5 main things that we check. So we check the volume, the concentration, the amount, and the quality, as well as something which I've called plate integrity. So the quality is the trickiest part to measure, since there are so many different factors that can interfere with library construction. As Pavel showed you a few tables with different kinds of interference factors. So for genomic DNA, we will ask you to run an agarose gel to visualize the integrity of the DNA, because degraded and nicked strands will not amplify in amplification-based library preparations, and it will also not cluster properly on the flow cells, even if we use non-amplification-based preparations. And it is a rather crude method of measuring quality, we're aware of that, but so if you see a big smear like this on the gel, it could depend on a number of factors. I mean, in this case, it looks like in these cases here, it looks like it's a lot of RNA contamination. But it could also be that the samples are degraded somehow, and this will definitely affect the library preparation. And also for RNA, we run it on a bioanalyzer or the caliper instrument to look at the RIN values, as Pavel also mentioned before. So the volume, concentration, and amount, these are the easier things that we can measure. However, the concentration might vary quite a lot depending on what method you use, as Pavel also mentioned. And we also recommend using picogreen or qubit-based or fluorescent-based methods, since these are more exact and not so prone to deviate depending on contaminants and so on. And there are several reasons we need to set limits on volume, concentration, and amounts. So our liquid handling system has a minimum pipetting volume where the accuracy is still acceptable. So this basically defines a maximum concentration of the sample. And there are also limits set by our quality control methods to make sure that the samples are within the detection limits of the methods that we use. All library preparations also have a set sample volume, of course, and an optimum amount for producing a good library. So this basically sets a minimum concentration of the samples that we have. And what I call the plate integrity check, this normally does not really affect the sample QC, but still an important part of the quality control. So we will send out barcodes, plates, and seals for each project after the project has been accepted. And this is because our robots, they have been calibrated to this particular plate type that we send out. And we also know that the particular seal that we give you works in all of the temperatures we use for storing samples. And the seals are also thin enough to allow piercing by pipetting, which is required for some of our protocols. And we also check for things like seal adhesion to make sure that no spilling over has occurred. And we also check that the sample positions in the plate corresponds with the information that you send us. Because sometimes we can notice that there are volume in wells that has not been designated a sample, and this could mean that there has been some problems with or errors in the sample loading. And so finally, so what can you do to make the quality control process as easy as possible? Well, first of all, don't hesitate to contact us early in the experiment to discuss issues or questions about quality control and the sample preparation and so on. And you should also measure the samples and make sure they are adjusted to the requirements that we give out. And you will add this. So this is a summary of the information, the sample-related information that we ask for different types of samples. So you write down this information in an Excel sheet and send to us so we can review it before the samples are submitted. And so, I mean, this way we can address possible inconsistencies so you can correct them before the samples are finally submitted to us. And this was basically it for me. So I'll be happy to take any questions. So I haven't taken any specific things about our prep. So if you have specific questions about requirements for particular samples, I'll be happy to answer that for you. We can also discuss during the coffee break or after the seminar. Right?
So, I was wondering, so you want to prepare the library yourself? You do not accept that?
I did not mention that, but of course, if you want, you can prepare your own libraries. And we will also make a quality control of that, similar to our internal quality control when we do libraries ourselves. Checking the concentration of the library and size distribution and so on.